Okay, today we're going to cover the nervous system. So this is Bio 168. This will be material covered on the third exam. First thing you want to do is always remember in anatomy you always have things called functional units. So in the nervous system we're going to look at what's called the functional unit of the nervous system being the neuron. So if I draw kind of a generic looking neuron looking under an electron microscope, I'm going to see that there are certain structures that we're going to be looking at. So if you notice that the first structure I've got listed here is an axon. So the axon is actually that extension that goes from right about here, comes out, and extends to about there. So let's define this word axon first. It is a long cytoplasmic extension of the cell body. And again, the cell body is the soma. So therefore, if you notice, this axon is connected or an extension of this cell body. So that's why we've got the word cell body. Of course, that cell body is also called the soma. And then if you notice, there's some small or shorter extensions coming off of that cell body. I see one here, and I see another one over here. And therefore, those are called dendrites. So therefore, a dendrite is going to be a short cytoplasmic extension of the cell body. And then as I look inside of this cell body, I've got an N written in the middle, and just like any other cell, it has to have an organelle that's going to control the cell. So therefore that nucleus is going to be contained in the cell body. Of course, that's going to be an organelle, which just means little organ. And then the other significant structure that you're going to see inside of a neuron is the one that I drew here, is with the squiggly line on the inside, called a nissl, N-I-S-S-L body, a nissl body. The nissl body is significant because that's a protein. Remember, protein is the acronym C-H-O-N. A protein synthesis organelle. And then when I look at the axon itself, you notice I drew these bumps running over the top of it, and therefore I describe them as being myelin. So myelin equals insulation equals white fat. So that is what's going to insulate that axon, just kind of when you think of like a wire has insulation around it. So again, looking at the structure just very quickly, we've got a cell body, just like any other cell in the body has. That cell body's got certain structures. It's got the nucleus, which is an organelle on the inside, a nissl body for protein synthesis. It's got long extensions that are generally covered with myelin as insulation, and it's got short extensions that are called dendrites. Now we want to take a look at that same structure and just understand a little bit more about an axon. So I'm just going to draw an axon, and I'm going to put those same bumps over the top of it. So that is a axon. What do we know about an axon? Well, we know that we're going to draw an arrow in this direction. And since the cell body is seated over here, or located, this is going to be sending impulses away from the cell body. So we know that an axon sends impulses away from the cell body. And therefore, these bumps that I put on here, which I've already described to you and say are called myelin, are going to insulate that. And that's why we said that an axon is long. The longer a wire is, the longer an extension is, the more insulation you have. There's a saying that goes along in anatomy and physiology that distance equals resistance. So to make you understand why we need to know about this myelin, think about an electric line. So here's an electric pole, and let's just say this is in Wilmington. And let's say that it has a wire that's extending all the way to Wrightsville Beach. And then, of course, running between them is this wire. Well, if I started out in Wilmington with 110 volts, and I ran that all the way to Wrightsville Beach without insulation, so a bare wire, by the time it got to Wrightsville Beach, it would probably be down to about 20 volts. So the distance equaled resistance. So what I do if I'm talking about electricity running between two towns on wires, I add insulation. So in order to go a distance without losing voltage, I add insulation. Well, of course, if we were talking about wires in a township, naturally those wires would have a plastic or some type of, that used to be asbestos covering over it or cloth covering. But in the human body, it replaces that with white fat or myelin, or the myelin sheath. Okay? So therefore, that's where you get that term white matter. So when I now add this insulation to this wire, if I put insulation on the wire, comparing it as an analogy to the human body, if I started out with 110 volts, I would end up in Wrightsville Beach with 110 volts. But the other issue you have is that even when you insulate a wire, you have to somehow keep that voltage up. So you're going to see as you're driving along the road that there's going to be voltage regulators along the way. And we'll just write here the term of voltage regulator. The importance of that is to keep the voltage constant when I have that distance all the way from Wilmington to Wrightsville Beach. So now if we take that same analogy and extend that now to the human body, we're going to be looking at that same structure again, which is the axon. We're going to put the myelin sheath over the top of it. So again, that axon is nothing more than that wire that's running between Wilmington and Wrightsville Beach. And so remember, running here, the electrical impulses are running away from the cell body. And so every time that voltage runs down this axon, it's going to jump across each one of these nodes. And those nodes and that jumping across is called saltatory conduction. And each one of those nodes, and those nodes are called, those little indentations, are called the nodes of Ranvier. And they're really analogous to those structures that I had when I was talking about the wire running from Wilmington to Wrightsville Beach. So if you just back up to the page before that, and you notice that I put this voltage regulator in here, I could also say that that voltage regulator is really analogous to that structure called a node of Ranvier. So in that node of Ranvier, which is going to act as a regulator, I'm going to have a depolarization and a repolarization, just like when we talked about in the heart. And that's what's going to keep that current and that voltage constant. But now instead of talking about 110 volts or 10 volts, we're talking about millivolts. Small incremental electrical energy units that are running all the way down that axon, all the way connecting it either to a nerve or connecting it to another axon or connecting to a dendrite. So again, that's the significance of knowing that. So that's why for this, knowing these nodes of Ranvier, we have to always consider our electrolytes. So that's why electrolytes such as sodium and chloride and potassium and calcium are so significant, not only in controlling our skeletal muscle, not only in controlling my cardiac muscle or my smooth muscle, but also in controlling my nervous system. So now if we know that a little bit about how this actually is working with this concept of saltatory conduction, first thing we want to also talk about is the different types of neurons. And I could spend hours and hours talking about the nervous system. But just think that if I've got one axon and one dendrite, it's going to be called a bipolar neuron. Two poles. It's got an axon, it's got a cell body, and it's got a dendrite. And of course, I'm always going to be putting this on here, this myelin sheath, because generally it's going to be that myelin sheath that's going to make this white matter. So just again, to add to this, just remember that axons are generally myelinated or insulated, and cell bodies and dendrites are unmyelinated. That's why we're going to say that axons are going to be white and cell bodies and dendrites are going to be gray. So white matter, gray matter. That's where the terminology comes from. So now if I now just took and said, well, there's a one axon cell body, and this one has got two dendrites, we're going to say that that's a multipolar neuron. So multipolar neuron is going to be one axon and two or more dendrites. And just think that, remember, there's billions of these inside of the human body. The brain, some people say, some books will tell you there's 10 billion, 20 billion, 30 billion. I don't think there's any exact number of how many neurons there are inside of the brain. But also realize these neurons are also making up all the nerves of the body. And so then we've got, as a general rule, we've got bipolar neurons, multipolar neurons, and then there's another one that's going to be called a unipolar. And a unipolar neuron is just going to have one axon. And so therefore, it's just going to be this structure connected to a cell body with the myelin sheath. And then we've got a fourth type that's a pseudo-unipolar. Remember, pseudo is false, pseudo-unipolar. And just think of that, it looks like it's only got one pole, but it really has two. So it's really got fused poles. So don't, as I say, don't lose a lot of sleep about knowing all that, but just realizing that as a general rule, most of the neurons in my body are going to be multipolar. So now that we know a little bit about what I would call the microscopic anatomy of the nervous system, then we've got to have something also called a structural unit. So since the functional unit was the neuron, the structural unit was the neuroglia. And sometimes that neuroglia are called glial cells. So neuroglial cells or glia cells are going to be structural. They are going to also be mitotic. So when you think of the neurons, as a general rule, I always use that word general rule because there's always exceptions to the rules, is that structural units or neuroglia are mitotic, and I'm going to use the word generally because there are some exceptions. But I could say versus neurons, which are generally amitotic. That's why when people get brain cancer, you'd say, where is the cancer? Well, the cancer can only be in a cell that has cell division, because cancer is really defined as uncontrolled cell division. So therefore, I can only really have cancer in a cell that can undergo cell division, and therefore that would be a glial cell. So now let's just think of a couple types of glial cells that are significant. If you go to your book, it's going to go through all the different types that there are. We're just going to, for right now, because these lectures only can structural kind of tell you the high points or the significant points of the material, is think that an example would be something called an astrocyte. Well, astrocyte, again, this is a neuroglia. It gets its name from astro, which means star-shaped, and cyte means cell, a star-shaped cell. It's going to have a function. It's going to bind blood vessels to neurons in the central nervous system. So therefore you say, well, how do I really draw something like that? Well, just kind of think that if this is a axon sitting over here, and this is a cell body of a neuron, and there's a dendrite, I've got to somehow provide nutrition to it. So these cells are going to be star-shaped, so it's going to have points on it. That actually is a astrocyte. And it's actually connected to a blood vessel. So the blood vessel is going to be delivering oxygen as a nutrient gas, CO2 is a waste gas, and of course sugar. And it's going to bring in that sugar and that nutrition and that oxygen, and it's going to be bringing it through an astrocyte, the star-shaped cell, and keep my neurons alive. So therefore, this is kind of a common tumor people develop called an astrocytoma. Why is it significant? Well, just think of it. If someone had cancer somewhere in their body and it somehow got into the bloodstream, it would carry in the bloodstream to the blood vessels of the brain. And so I may start out with breast cancer or melanoma, but because the systemic circuit would bring those cancer cells from the melanoma that might be on my back into the bloodstream or systemic circuit going to my brain, those cancer cells would transfer over to my astrocytes and I would develop an astrocytoma. So the astrocytoma is a common brain tumor. Okay? So that's one of them. Now, if I was, remember, this is in the central nervous system. So if I was in it's an astrocyte in the CNS, but the cell that's analogous to it, or basically that does the same function in the peripheral nervous system, is going to be something called a satellite cell. So an astrocyte is called a satellite cell in the PNS, or the peripheral nervous system. Okay? So that's one of the cells we want to know about, astrocyte. Let's think of another one. We said that we've got myelin. Remember, myelin equals insulation. Well, I've got to produce cells that make myelin. If I'm in the central nervous system, I produce myelin or insulation or white fat with a oligodendrocyte. So when I'm drawing that cell like this, and I'm saying that this is covered with myelin, that insulation in the central nervous system is going to come from an oligodendrocyte. But I also have to produce myelin in my peripheral nervous system. So in the PNS, it's going to be produced. So myelin in the PNS is produced by a Schwann cell. So same thing. I've got to have myelin both in my central and my peripheral, except it's an oligodendrocyte in the central, but it's called a Schwann cell in the peripheral. So again, just think of the significance. And again, I'm not going to go through every one of the different types of cells that are called glial cells in the body, because those are covered at length in the book. But if you kind of start differentiating between ones found in central and ones found in peripheral, and understand the significance that when you get something that carries a blood supply, that's how a tumor can become metastatic. So the next thing we want to do, since we know that that's the overview really of the functional unit and the structural unit of the nervous system, let's differentiate what we would call what is the CNS. So CNS, remember, is the central nervous system. And whether you were taking a board exam or some type of a review exam in any type of health science, you're always going to see a picture or a diagram that really looks like this. And therefore, what that is telling me is I'm really looking, and I'm just going to draw a couple little structures in here so you can kind of get the big picture, as I call it. You're really looking at this up here, which is the brain. So the brain is in the central nervous system. I know that this region right in here, so if this is the foramen magnum, or which is the largest opening in the skull, in the cranial vault, this is actually going to be the other portion called the brain stem. And then from there down is going to be the spinal cord. So the big picture is the central nervous system is comprised of the brain stem, and the spinal cord. But it also comprises, if you recall, when we were talking about the bones, remember there was CN1 and CN2. CN1 is, remember, that is the olfactory nerve, and CN2 is the optic nerve. Those nerves are both sensory, but remember, they're always CNS. So when someone says to you, what's in the central nervous system, the big picture is going to be the brain stem, the spinal cord, CN1, and CN2. And again, just to refresh your recollection, remember CN1, the olfactory nerve, we're always going to associate that with the ethmoid bone. And CN2, we're always going to associate with the sphenoid bone. But now we just have to divide up these large structures into a couple more smaller structures. So just remember, when someone uses the word brain, the brain is really comprised of the two cerebral hemispheres, and those are called the cerebrum. It's also going to be comprised of the cerebellum. Sometimes that's called the little brain, and I'll kind of draw that for you in a minute to kind of show you where that really is located, towards the back of your head, somewhere by the occipital bone. When I'm talking about the brain stem, that's going to be made out of the pons, the medulla, and the midbrain. So those are your portions of your brain stem. Remember, those are what are called the vegetative brain. And then finally we have the spinal cord. Always think of the spinal cord as approximately 18 inches long. So again, brain, two cerebral hemispheres making up the cerebrum, brain stem comprised of the pons, medulla, midbrain, and then the spinal cord approximately 18 inches long. That was what would be considered my central nervous system. Before I move on to the peripheral nervous system, I just want to kind of tie up some things that I think are significant to know, that are kind of high points of material that's inside of the book. So when we're talking about, for example, this brain stem, just remember that the pons and the medulla are going to be responsible for heartbeat and respiration. And that's why we get that term that the brain stem is called the vegetable or the vegetative portion. Vegetative. The vegetative brain. Back in 1975, the Karen Quinlan case really set the concept of brain death. And brain death really looked at someone that was a girl that took an overdose of some medication. She comes into the emergency room in a coma. They do a electroencephalogram, which comes back a flatline EEG. They wanted to turn the ventilator off. It ended up all the way to the Supreme Court because they said that just because someone has a flat EEG does not mean that they're dead. It's just saying that they're brain dead. So it went through a series of variations of how you determine brain death by doing cranial nerve reflexes and basically using dye to look at the blood supply going to the brain, and that eventually went on for years in the Supreme Court. And then, of course, when they shut off the ventilator, she started breathing on her own, and then the question became of whether you can put a feeding tube and disconnect the feeding tube. And that case went on and on and on. Same thing happened and went to the Supreme Court on the Chavo case in Florida. Same issues that became again, because it was a question of whether the family wanted to shut the ventilator and the husband wanted to keep her alive. Well, eventually this court said they could turn the ventilator off and stop all life support, and when they did the autopsy of the brain, it was nothing more than water coming out. But why she was able to either breathe on her own or have a heartbeat on her own is because these are the vegetative brain, and that's where that word came, kind of a slang term, that someone is in the hospital in a coma or a vegetable or a vegetative state. So that's how you want to think of the significance of knowing those structures, pons and medulla. Now, since we know that we've got this brain, we know that the brain has got covering. So the brain and the spinal cord are going to have coverings. So those are what are called the coverings of the brain stem, and the spinal cord. And therefore, just think of it as a circle. So if we use the cranial vault as the best example, just remember that I've got my bone over here. So this is bone. Below the bone, I'm going to have a space. So since this is bone, and this is going to be that one of the meninges called the dura mater or tough mother, this space between the dura mater and the bone is going to be called the epidural space. If you're a female and you've had to have a spinal anesthetic or an epidural, that's where they would inject it into the thecal lining of the epidural space. Below the dura mater is going to be another one of the meninges, and that's going to be called the arachnoid mater, or the spider web. Arachna is a spider. That's going to be all these blood vessels, hence it got its name arachnoid. But again, you're going to have a space between the dura mater and the arachnoid called the subdural space. Now below that, below the arachnoid mater, there's going to be another covering, another of the meninges called a pia mater. Pia mater is almost like tissue paper. But again, because this is the arachnoid mater, that's going to be the subarachnoid space. And now below the pia mater is where I'm going to see the surface of the brain. So when I think of the surface of the brain, which that's what this is, so this is my brain, I'm going to see, notice that brain is going to be much larger than it is, but it's compressed because it's got elevations and depressions. And you kind of think of that analogy to cristae. The cristae are those folded membranes on the inside of a mitochondria. And so therefore, these elevations are like this, and this is a depression. So the elevation is going to be a gyrus, and the depression is going to be a sulcus. So those gyri, which is plural, so the gyri, G-Y-R-I, which is just plural for a gyrus, or the sulci, plural for a sulcus, are what going to compress my brain and give that elevation and depression surface that you see when you open or take off the calvaria. So just remember that it's the significance of all of these is someone hits you with a hammer. And when you get hit with the hammer, you get bleeding or breakage of the bone. It's going to cause a epidural hematoma, bleeding into the epidural space. If the person hits you harder, now you're going to have bleeding into the subdural space. And of course, if it's a significant blow, the significance here is that the subarachnoid space is going to contain CSF, or cerebral spinal fluid. And it's that cerebral spinal fluid that you'll be able to do a spinal tap and see a spinal tap in the lumbar region, or LP, or a lumbar puncture. If I have blood in the lumbar puncture cerebral spinal fluid, that means it's going to be positive for the same thing inside of my subarachnoid space, because it's a continuous flow. So same thing is going to be with the spinal cord. You know, when we talk about the spinal cord, you probably recall when we did bones, we drew something like this, and we said, this is really a generic looking vertebra. And if I look at a generic vertebra, remember I had a spinous process, and I had a transverse process, and then remember this was called the body, or the centrum, and then there is a opening right about here, and that's called the spinal canal. Sometimes it's called the vertebral canal. And then there's a notch that runs really right inside of here. I don't want to put it here. I want to kind of stick it really right here. So I'm going to stick a notch right in here. And we're going to call that the inter. Remember, that means between. Vertebral, a vertebra, foramen. And remember, sitting on the top of this body, or centrum, was my IVD, or my intervertebral disc. So if I now say, well, where's my spinal cord? It's actually sitting right inside of here. And I'm just going to draw it real quickly just to show it. So that structure sitting in there is my spinal cord. But we can continue that same story of the bone that covers the brain and the meninges that cover the brain as the bone and meninges that cover the spinal cord. So now, same thing. So if I was to draw your spinal cord, here's my spinal cord that's sitting inside of there. It's going to have that H pattern in the middle. And that's going to have a little opening like this. And then surrounding that are going to again be these coverings. So let's identify this, what we're looking at. Well, we know that this H pattern that I see in the middle is going to be gray matter. And the area surrounding it that's not shadowed is actually going to be white matter. That's telling me that the white matter is myelinated or insulated, and the gray matter is going to be unmyelinated or not insulated. And then sitting in the middle of that is something called the central canal. And you recall when I said that the subarachnoid space is going to have cerebral spinal fluid, well, that central canal is also going to contain CSF. So if this is my spinal cord, it's going to have coverings just like the brain did. So if this is my spinal cord, remember the spinal cord is going to have a covering, so that first one is going to be the pia mater. Above the pia mater is going to be the arachnoid matter. Above the arachnoid matter is going to be the dura mater. And above the dura mater is going to be bone. So the exact same coverings and exact same spaces. If this is the dura mater, this is the epidural space. Therefore, again, woman has a epidural anesthetic for a delivery of a child. Epidural space is where I'm going to do the anesthetic. If this is the dura mater, then this is going to be the subdural space. If this is the pia mater and this is the arachnoid mater, this is going to be the subarachnoid space. Again, remember the subarachnoid space has CSF, cerebral spinal fluid. And finally, that pia mater is sitting right over the top of that spinal cord itself. Now we can tie this really all together very quickly and just saying that if we draw that large circle one more time, and we draw that H in the middle, and that central canal, and coming off of there are going to be roots. So there's going to be a root coming off of the top and the bottom. Remember, this is going to be gray matter here that's unmyelinated, and that's going to be white matter myelinated. I'm going to have a sensory root. I'm going to have a motor root. Same on this side, a sensory root and a motor root. And therefore that sensory root and that motor root will come together, and they're going to pass through that opening called the intervertebral foramen. And they're going to come out of the spinal cord as a spinal nerve. So now if we just tie that together, if this again is going to be my vertebra, and here's my spinal cord sitting in the middle here, coming off of here are going to be those roots that will join together, and they'll come out of that intervertebral foramen on either side. And that's why when we look at this same picture that I said, that you're always going to see on exams, you're always going to see something that looks like this, and we're going to say, well, remember, that's the brain up here. That's my foramen magnum. That's my brain stem. And therefore I said this is my spinal cord. And you're always going to see coming off of that spinal cord, and I'm not going to draw all of them in here, you're going to see these 31 pair of spinal nerves. And those 31 pair of spinal nerves are going to be all PNS, they're peripheral nerves, and they're all mixed, which means they're motor and sensory. Because all they really are is the joining of the sensory root and the motor root exiting the intervertebral foramen as a spinal nerve. So that's the big picture, I would say, of the brain, brainstem, spinal cord, and then thinking of the structures of a neuron and neuroglia and the concept of how we send impulses from the brain or the spinal cord to a muscle. So, if we'd say another simple concept that you want to really understand is something called the neuromuscular junction. Sometimes that's called the NMJ, or it could be called the myoneural junction. Just two different words for the same thing. So that's the NMJ, or neuromuscular junction. This is the myoneural junction, which is the MNJ. But no matter what you really call it, all it's really talking about is how somehow I'm getting a nerve attaching to a muscle, motor end plate. So let's just real simply say, here's a nerve coming down, and this is a muscle. And where it attaches is going to be called a motor end plate. And the real simple way to understand this is really to say, well, how do I make this really understandable and not complicated? Because I know when I was taught this, it just became a bunch of jumble the way it was taught. But if I think of it very simply and say, well, here's a finger. So let's just say this is my index finger. And here's the fingernail. And let's just say that sitting inside of this finger is a muscle. And since I'm saying this is the index finger, and I want to move this muscle, and let's just say the name of that muscle is the extensor indicis. And let's just, for simple purposes, let's just say that sitting below the end of this finger here is a candle. So this is a candle which is producing heat. So there must be a sensory receptor sitting right here. So that's going to be called a sensory or afferent or ascending receptor. So I took my finger and I put it over the top of this candle, which was producing heat. Well, my muscle is going to say, wow, that finger is, and that receptor says, I need to get that finger moved away. So I'm just going to draw a real simple picture here of the brain, stem, and spinal cord. And remember I said there's spinal nerves coming off of here, and I'm just going to draw one of them just for today's purposes. And I'm going to say that's a spinal nerve, an SN. Remember, spinal nerves are going to have two pieces to it. It's going to have a motor root, and it's going to have a sensory root. So that heat is going to transfer to this sensory, afferent, ascending receptor, and it's going to travel in the finger, in the peripheral nervous system. It's going to go up through my shoulder, up into my arm, and it's going to enter my spinal cord through one of the spinal nerves. That's why it's ascending. It's going to travel up my spinal cord into my brain. Now I'm inside of my brain. If you notice that I'm now on my right side of my brain, my right side of my brain is going to have to process that information, and it's going to have neurons that are connected to the right side of the brain to the left side of the brain, and my brain hopefully is going to say, Mark, you better move that finger away from that heat source or it's going to get a burn. So it's going to have interneurons or connecting neurons to the left side of the brain. It's going to send that down the brain. And so now that's going to be called a efferent or motor or descending neuron. And it's going to travel to this motor root of that spinal nerve, and that motor root of that spinal nerve is going to track along with this one, except it's going to end up at the muscle. And therefore it's going to tell that muscle to move away. And it's going to take that extensor indicis and cause it to extend and move that finger away from the heat source. So again, if you just think of this, it's very simple. Just think of this as how the human body acts, except if it's something like the Babinski reflex or the patellar tendon reflex. Those are reflexes. Reflexes are going to the spinal cord. But this is how basically 99% of your body functions using the motor and sensory receptors. So I'm going to have a heat source. I'm going to have my finger here, and the finger's got a muscle inside of it called the extensor indicis. The heat source is going to tell this sensory or afferent or ascending receptor, which is nothing more than really a neuron. Remember, nerves are made out of neurons. It's going to send that ascending, afferent sensor receptor through the sensory root of the spinal nerve to the right side of my brain. The right side of my brain is going to process that and say, you better move your finger. It's going to have an interneuron that's going to send that information over to the left side of the brain. The left side of the brain is going to then send an efferent or a motor or a descending neuron down the brain down to the brainstem, down to the spinal cord. It's going to exit out that same spinal nerve except out of the motor root. The motor root will then pass alongside of that nerve to the motor nerve going to the extensor indicis muscle, and going through that motor end plate is going to cause a depolarization and repolarization and cause that muscle to extend and move away from that heat source. And that really is how it's going to work. And so that's the only difference between that is that versus a reflex. So just think of reflex actions, or when you're basically, the best one to think of is the patellar tendon reflex. And that's also called the knee jerk. And I'm not going to draw a knee here, but if you just kind of think of it, that I'm thinking all of you have probably had your deep tendon reflexes checked. And if you were to sit over the top of a table and I would tap just below your patella, I would tap the receptor that's there. When I tap that, it's going to send a afferent or ascending or sensory receptor to my spinal cord. My spinal cord is going to say, I need to move that muscle away from that impulse. So if I've got good tendon reflexes, my spinal cord is going to send out a descending or motor or efferent neuron to the muscle, and it's going to cause my knee to jerk out, my leg to jerk out. And so you kind of think that if my knee is going to jerk out, I'm going to be extending my leg, and therefore that's going to be my quadriceps femoris or my quads. So that's really the distinction between a reflex and a normal arc of the brain, brainstem, and spinal cord to an afferent receptor. Okay? There's another one that's significant you've probably heard about called a Babinski reflex. Babinski reflex is really going to be significant in an infant. Generally, an infant is not fully myelinated till about 20 months. So an infant's got a positive Babinski reflex up to about 20 months. But suppose I've got a person that's 35 years old, and you do reflexes on them and they have a positive Babinski. The first thing that comes to mind is the disease called MS, or multiple sclerosis. So we can tie all that together that we've so far learned today by just thinking of one pathology. Everyone here certainly has heard of the pathology called MS. All right, multiple sclerosis. We want to think of a pathology all of us have heard something about. Remember, MS is an autoimmune disorder. Remember, autoimmune is really defined as recognizes self as non-self. So if I want to make a very simple discussion of understanding MS and tying all of this together, just think that if I was looking at a neuron, and here's my axon, and we'll just draw a multipolar neuron. Remember, the axon in a normal person is covered with the myelin sheath. And if you think of MS, how it usually presents itself, and I talked a little bit about that when we did bones, and I told you that MS often presents itself. So the presentation of MS is oftentimes optic neuritis. Optic neuritis means that your optic nerve, cranial number 2, is kind of retinal flashes you would get. So you'd be driving at night and you'd see almost like lightning flashes in the periphery, but then you'd close the right eye, still see them. You'd close the left eye and still see them. You'd close both eyes and still see them. So therefore, that would be a retinal flash. So the diagnosis there, just with that, would either be, maybe you've got a brain tumor, maybe you've got a pituitary adenoma, but the bad side of it could be MS, multiple sclerosis. The other presentation you have of it, besides optic neuritis, would be foot drop. You're going to flex and extend your foot as you're walking, and all you'll notice when you go to bring your plantar surface down or push your toes down, and basically remember standing on your toes is going to be plantar flexion, you notice your foot is just floppy. It really almost seems disconnected and you almost trip or fall. So those are two common early presentations of MS. And so therefore, when you think of what MS would be, if I was to look at a neuron of a person with multiple sclerosis or something where I've got a degeneration of the myelin sheath, if that was a normal looking axon with the myelin sheath and the nodes of Ranvier, if I had MS and I did pathology and did a biopsy, I would notice that I'm missing some of the myelin that's connecting. So the electrical impulse, remember, that passes down, well, it got to here, so there was a delayed conduction here because there was no insulation. And then it did its saltatory conduction here, and then it moved along. But then there was a delay because I have no insulation here. And because I've lost that insulation, that's why I have the foot drop or the jittery movement or the optic neuritis that I would see. And the same thing I'm seeing here. So what has happened in that person is he's lost his myelin sheath. But an autoimmune disorder, remember, self recognizes self as non-self, so my body is literally destroying its own myelin sheath, and therefore I'm presenting with the pathology of multiple sclerosis. I know that the special senses are very complicated and detailed. Nobody totally really, understands all the senses, especially smell. I think it's very significant to memorize the 12 pairs of cranial nerves. I think that you always want to remember that the mnemonic, and there's a lot of mnemonics in the back of the book you'll see, and I always say, on old Olympus, towering, top, a French and German, viewed, some, hops, and I think you're all familiar with hops because that's what they make beer out of. And so that's a mnemonic that you can memorize. There's a lot of mnemonics in the back of the book, so don't feel that you have to be stuck with that one. Just remember that's really telling me that O is going to stand for olfactory. The second O is going to be optic. We've already talked about that. The third is the oculomotor. Remember we learned about that and we talked about the muscles of the extrinsic eye and the intrinsic eye. The T is going to be the trochlear. Remember the trochlear we talked about, SO4, LR6, the rest are 3. The next T is the trigeminal. Trigeminal means it's got 3 branches, so therefore trigeminal. A is the abducens. Remember that's going to be 6, so if you just put a number 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Abducens, remember that again. Just think back, SO4, LR6, the rest are 3. Therefore, that's your lateral rectus. F is going to be your facial. Remember, that's going to be all your muscles of facial expression except the LPS or levator palpebrae superioris. A is going to be the auditory. But remember, the auditory has been changed, named. It'll also be called the vestibulo-cochlear. So therefore, think of the hearing and balance. G, glossopharyngeal. Remember, glosso is tongue, pharyngeal is pharynx or throat. V is vagus. Remember, the vagus nerve is going to control your heart. So you always think of the vagus nerve control your heart or the sympathetic nervous system besides that. S, your spinal accessory nerve. If you recall back from the muscles, remember the spinal accessory nerve innervated the trapezius, sternocleidomastoid, and H is hypoglossal. And remember, hypoglossal is going to be, again, your hypo below the tongue. And then you want to memorize another mnemonic, and again, you can use whatever one you want. This is one that a lot of people use, and let's say some, say, marry, money, but my brother says big, and we'll just use big brains for today. There's different terms. Matter most. And so S stands for sensory. And M stands for motor, and B stands for both, which means it's mixed motor sensory. So therefore, if this is 1, that's sensory, 2, sensory, 3, motor, 4, motor, 5, B both mixed motor sensory, 6 is M motor, 7, B both motor sensory, so therefore mixed, auditory, number 6, sensory. Auditory makes sense because hearing, balance. Glossopharyngeal, B, both, mixed motor sensory. Viewed, vagus, mixed motor sensory. 11, motor, and 12, motor. And then you just want to kind of keep some generalities in your mind. Remember, when you're thinking of intrinsic, extrinsic eye, remember you're always going to think 3, 4, 6. When you think swallow, think of some generalities of 9, 10, 11, 12. When you think of chewing or mastication, you're going to think of 5. When you go on your review sheet, you're going to see that each one of these nerves is laid out specifically as to what they innervate and their significance. So those are things that you're going to want to memorize and know for that third exam. The last thing I just want to go through for the nervous system is just a quick look at the eye. We've talked about the eye before when we talked about the muscles. So I think that's always important to be able to draw an eye and label an eye. So I'm going to take an eye, I'm going to start out here, I'm going to have a structure on the front. I'm going to have a covering that kind of covers a portion of the eye here. And I'm going to have my eyelid coming across here. I'm going to have an opening and a muscle here. I'm going to have another muscle and a structure. And then I'm going to have a inner side layer that's going to pass to the outside of the eye, a middle layer, and an outer layer. So here's the outer layer of the eye. That's your sclera, or the white of the eye. This darkened layer that I'm going to just shade in here is going to be the middle layer, or the choroid layer. That's going to have my melanin significance. That's going to, just like you think an albino, that's going to be pink. Hence, they have no vision basically in bright sunlight. I'm going to have my inner layer. That's going to be my retina. Remember, retina is going to have my photoreceptors of rods and cones. This is cranial nerve number 2, or my optic nerve that's afferent, that's sensory, that's always CNS, that's going to my brain for vision. This is going to be the muscle that's going to focus my lens, so therefore that's going to be the ciliary muscle. This is going to be my lens. I always think of the lens as a consistency of a silicone or water saline filled breast implant. And of course it hardens with death, but if you saw in a living structure, that's how it's able to move so rapidly and constantly focus. Therefore, I'm going to have this opening here, which we've talked about before, being the iris. That's your pupillary light reflex we've discussed. So therefore, what's going to control the iris is going to be the muscle. So that's the muscle that controls it, and that opening that we see. So this is the iris we're seeing on both sides, and that opening is going to be the pupil. And therefore, that clear portion of the eye is going to be the cornea, and therefore the outer layer of the eye, the outer layer of the eye is, so this is the sclera, this is the retina, this is the choroid, and so right across the front, covering the anterior portion of the sclera, is going to be the conjunctiva. So the conjunctiva is what's going to get red with pink eye, and therefore that's why it's called conjunctivitis. So if you kind of think of the big picture, that's how it's going to work. And again, you're going to want to remember a couple terms. I'm just going to write them down for today. And again, go to your book, make sure you do all the end of chapter questions and review all this, but you'll see it's fairly interesting, but it's just the one that's very complicated. It's really the ear. The ear really lends itself to really memorizing and knowing the diagram and going very carefully through the book. But when I think of the eye, remember you've got hyperopia, you've got myopia, you've got emmetropia, and you got presbyopia. So that kind of summarizes the nerves and special senses, and now what you're going to want to do is go back to the book and go through that chapters 7 and 8, and those are really going to focus on the nervous system and the special senses.